Okay, if you're watching this to get credit, as usual, you're going to pay attention throughout the lecture. I'll give a series of attendance words or attendance phrases. You're going to write those down, and every time I say one, not only will you write down the word or the phrase, but also take a screenshot. At the very end, the last thing you need to write down, and there's no screenshot needed for this, is to ask me a question about the material presented in the video or the lecture, and then email it to me, and that's how you get your credit. So the first word for attendance, you to start it off, is going to be Zoom. Sort of like how I just zoomed in to that screen right there. So Zoom is the first word for attendance. You also need a screenshot for every word that I say, including this first one. So I should see a screenshot that shows the desktop there. And that's it. It is about 7:32. So I'd say if you're watching the video, you could probably go ahead and skip ahead about 25 minutes. And that's when the lecture will start.
Morning.
Morning. Morning. Morning. Morning. All right, a little less than 5 minutes. Go ahead and get the process started here. All right, Brumfeld is not here yet. Champion is here. Gross is not here. Gunther is here. Randolph Phillip is here. Zion Hill is not. Jackson is not here. Keys is here. Evans is not here yet. Sigmund is not here yet. Slaughter is here. Smith is not here yet. Stover is here. Tompkins is not here yet. Van Nest is not here yet. Nick Watson is online. Young is not here yet. Zweiback is here. Lawrence is not here yet. Wilson is not here yet. And did I miss anybody? All right, I know we still have like 4 minutes to change, but I'm going to go ahead and at least start the announcements and reminders because some of it's just on repeat. So I don't mind saying it before everyone else gets here because they've probably heard it before. For example, you can still, or not still, you can meet me online to get all the answers for exam 1, which will help you study for the final exam because after the final exam comes from the first 3 exams. So you're going to need to know those correct answers. And you get a 5% boost to your exam 1 grade for meeting me online and getting that feedback. And like I've mentioned previously, it's not like I'll just give you the correct answers. I'll also explain why your answer was incorrect and, you know, why this is the correct answer. And in some cases, there might be a mistake that we catch, and then you just get, you know, you can actually get those points back. Or if you can argue against something, like I know this was incorrect, but here's what I was thinking. You know, if you can argue against a wrong answer, I might give you the point. If you can defend it, that means you understand it, right? And that's the purpose of the test, is to make sure you understand these concepts. So, any questions about meeting me online for exam 1? All right, again, exam 2 is open. You can retake it until exam 3 closes, and that won't be until November 9th. So I highly recommend it. You know, it's open book, unlimited attempts, not timed. So you have nothing to lose. I would highly recommend retaking the exam too. Any questions about that? All right, lab this week, it's virtual. Since I'm so behind, I'm gonna do what I did last week, which is I'll be in here for lab. And if you want to be in here for lab, you can be too, but it's completely virtual. So even if you come in for lab, you're gonna need your own computer and you're gonna need to be working on it, and then I'll be here if you need me. So you decide if you want to come to lab this week or not. Again, it's 100% virtual. I'll be here if you need me. Any questions about lab this week? Okay, next week lab is also virtual. I don't know if we're gonna meet again. Yeah. What? Question?
What?
Go ahead. Question?
So we got to stay here to do the lab.
Correct. This week, you don't have to be here if you don't want to, but I will be here if you need me. What was the other question?
No, that was the only question. I was just trying to understand.
Okay. Somebody else say something then? All right. Anyway, next week's lab is also virtual, which means no pre-lab due this weekend. So again, one less thing to worry about. Any questions about that? Speaking of which, it's a good time to remind you, now that you're going to have a little bit of extra time as far as this class is concerned, maybe you should use that time to fix your old mistakes. Like if you missed an assignment, go ahead and turn it in. If you've been absent, you need to make up for those points. Email me. I can tell you which stage you've been absent, and you can go back and watch the videos and get those points. Yeah, if you have labs that have a very low grade, I would recommend fixing those. You know, obviously I would do triage, like the lowest lab grade, I would work on that one and then start working your way up. It's a good time to remind you too, according to the syllabus, all that's not accepted, right? According to the syllabus, late work isn't accepted. According to the syllabus, I don't let you make corrections. But unofficially, you go ahead and turn it in. The reason I say unofficially is because I know what's going to happen. At the end of the semester, a handful of you are going to start caring about your grades, and then you're going to turn in 10 assignments on the last weekend, and you're going to say, why didn't you grade it? It's because I don't have time if you wait to do that. So any questions about that? All right, again, to remind you, I'm here to help you. You don't have to do any of this alone. I mean, technically, you kind of have to do the exams alone, but not even then. I can even meet you online while you're taking an exam. That way, if there's any questions, you're not quite sure what they're trying to ask, you can say, hey, can you explain this to me? I'll be happy to help. On a related note, these are my office hours. Again, Monday, 9 to 10, Wednesdays, 11 to 12, Fridays, 9 to 12. That's when you're guaranteed to find me online and guaranteed to find me in my office. So if you need me, please let me know. And I'm more than happy to meet you outside of those office hours. But we'll just have to plan on it. And as some of you know, sometimes that's hard to do, but I'll try my best. Any questions about office hours? All right, again, attendance is mandatory, participation is graded. I won't do that yet because I'm gonna wait until 8:01 until everybody, it's officially too late. I will say this though, there's a fundraiser tonight at Buffalo Wild Wings. I think it's like 4 to 9. I forgot what exact times. It's on the Facebook group. It's on my Facebook profile if you want to check it out. If you go there, you'll get extra credit. I forgot how much extra credit I said. But, anyway, if you do go there, make sure you take pictures of you there as evidence. Or if you see me there, come over and say, hey, I'm here. So you'll get extra credit for that. Any questions about that? All right, it's 8:01. Really quickly. Take attendance one more time, and then we can get started. All right. All right, so Brumfeld is here. Champion is here. Gross is not here. Gunther is here. Both Hills are here. Jackson is not here. Keys is here. Perez is here. Sigmund. Yep, Sigmund's here. Slaughter's here. Smith is here. Stover's here. Tompkins is not here. Van Nest is not here. Watson, I think, is online. Young is here. Zweiback is here. Lawrence is not here. Wilson is here. Did I miss anybody? All right, let's get started. So again, participation mandatory, or attendance mandatory, participation graded. we're gonna do the same thing we usually do, or the thing that we've been doing now, which is, again, all the attendance words will be mandatory, even if you're here. So the first word for attendance is going to be, well, a phrase, name, George Wells. Let's do Beetle, his last name, George Wells Beetle. Interesting timing. Today's his birthday. And he is the scientist, well, one other scientist, who basically discovered that genes control enzymes. So all the stuff you're about to learn in this chapter that we're starting today wouldn't have happened without this guy. So the timing was pretty effective. And again, if you're in person, you need to send those words by 9 a.m. If you're online, you need to send the words by 9 a.m. If you're watching the video, you need to take a screenshot. So you should see a screenshot of me pointing at George Wells Beetle's name. And that's it. Any questions? All right. So again, to remind you about the big picture here. Exam 3 is mostly going to be about reproduction, right? So you learned about cellular reproduction. You learned about mitosis and meiosis, which are the two different types of cellular reproduction. Then you learned about genetics, basically, right? Inheritance, which is the product of reproduction. Now, what we're going into is molecular biology. So essentially, it's everything you've learned, but now we're going to look at it like we're going to zoom in, I guess you could say. Instead of just saying, oh, the chromosomes did this and the chromosomes did that, now we're going to zoom in and look at all the enzymes and the machinery that attaches to those chromosomes. And we're going to learn how DNA does duplicate itself in the S phase of interphase. We're also going to learn about the stuff I was just telling you about with Mr. Beetle, which is how we go from a gene to a protein, or in that case, an enzyme. And remember, an enzyme is our proteins. But anyway, yeah, that's a little bit out of date, that date right there. But anyway, yeah, we're now starting chapter 9, which is molecular biology. This PowerPoint is again based off the old textbook because I like the way the old textbook presented the information. So again, if you're following along closely in the textbook that we're using, you might find that it's a little bit out of order. Don't worry about it. But we will start with this. This again, that old textbook kind of talks about how this is relevant to your everyday life. I'm trying to remember what I've got notes here. Yeah, all life on Earth shares a universal genetic code. So your DNA can be used to genetically modify bacteria or vice versa. And that's a lot of what's going on right there. And you're going to learn a little bit about that today. So because the genetic code is universal, you can take genes out of one species and insert it into another. And that's a great example because, you know, there's a big difference between humans and bacteria, but we can still take human genes and put them into bacteria and have them produce stuff for us. And opposite, we could take bacterial genes and insert them into the human genome. I'm not sure why you would want to do that, but it is possible. And then this, obviously that's a cow. How many of you ever heard mad cow disease? It's been a while since that's been a thing, but yeah. How about chronic wasting disease in deer? Okay, if you hunt, you probably heard of that. But anyway, you're going to learn a little bit more about that on this chapter, understand how it works. This, I told you we'd talk about this again. I gave you an example of that in the last chapter. And I said there's going to be at least 2 more examples of it in this semester. Does anybody remember what this is? Not that I expect you to, but because that was just an example. But if I know like what these 2 things are, how they're different from each other, how they're the same. Any guesses? Oh, okay, that's a regular old red blood cell. There's your first hand. You might now want to guess what that is. And knowing that you just got an example from that on Monday, and there was a picture of it.
That's sickle cell disease.
Yeah, perfect. Yeah, sickle cell disease. So you're going to learn how if we change one letter in your code of DNA, your A's, your T's, your C's, and your G's, you change one letter, and you go from this to that. And as you know from the example in the previous discussion, that one change makes that whole thing have that weird shape. And because of that, you just have this long list of symptoms and negative outcomes from the disease. So any questions about this slide? All right. I also saved this, even though I'm not going to talk about it and I'm not going to test you on it. But again, the old textbook had at the beginning of every chapter, it had like a news story, something that was in the news recently, or back then, where you could be like, oh yeah, I remember that. And then they're saying in this chapter, you're going to learn a little bit more about that. You'll have a better understanding of this news thing. So again, I'm not going to talk about it. You can read about it if you want. Zika was the big thing that came out right before this old textbook was presented. I bet if you were to get a newer edition now, I would assume they would talk about COVID, because if you were to truly understand how COVID works and how the COVID vaccine works and how mRNA vaccines work, that's this chapter. So it's a very important chapter as far as your everyday life is concerned. You're going to learn some important stuff. And again, like I mentioned before, as a college-educated citizen, hopefully, once you leave here, you're still thinking for yourself and you're doing research. And when someone says, hey, I think, we should do this kind of thing medically, then maybe you would think about, all right, well, let me look into that and see if I'm going to vote for this person or not, to see if I support what they're saying or not. But anyway, this chapter is going to be broken down in 3 main parts. Again, this is based on the old textbook. So same information, different order, different examples. But first, we're going to talk about DNA structure and replication, which you've already learned about the basics, right? Because that happens in S phase, interphase. So we're going to talk about that in greater detail. Then we're going to get the more complicated stuff. So we're going to learn how we actually go from DNA to RNA to protein. And as far as volume is concerned, quantity, that's probably the most important part of this chapter as far as the exam is concerned. More questions are going to come from that middle bullet point than the other two. And then finally, you're going to learn the basics about viruses and other non-cellular infectious agents. So here's another thing I like about this old textbook. Remember when you learned about Mendel, who's the father of genetics, the book kind of presented it as a story. So, hey, here's what Mendel was doing. Here's the timeline he was doing it. And they're kind of doing the same thing here. So again, I wanted to stick with it. It's telling it more like a story. Like, here's the timeline of what we used to know, of what we figured out, and when we figured it out. It's not really important that you understand these dates specifically, but I do want you to know the chronological order. Like, for example, the first thing we knew was that DNA was part of cells, right? In the late 1800s, someone figured that out. For an extra pen of paper or email, whatever, you can look that up. Who figured that out? Exactly what date did they figure that out? Send it to me in an email. Don't forget your sources. If it's just an email and you're just giving me the answer to the question, 1 point. But if you want to, you can write a paper about it, and then at which point the score would be based on basically how long your paper is. But yeah, you can look that up. So anyway, that's what we knew at first. We knew DNA was in the cells. We didn't know what it did. We didn't know its structure. We just knew it was there. And then in the 1930s, we figured out that there was one kind of molecule that is the basis of inheritance. And they hadn't put these two things together yet, right? So they knew DNA was in the cells. They knew that there was some kind of molecule that was in control of heredity in the cells, but they didn't know that DNA was it. And again, I'm not saying who, because it's not really important for this course, but you can look that up for extra credit. Who figured this out in the 30s? Anyway, then in the 40s, we learned a little bit more about DNA, right? We learned that these chromosomes are made up of 2 components, which you guys already know because we mentioned it before. Chromosomes are about half DNA and half protein, as you can see in these pictures. And to remind you, right, the DNA wraps around those histones. And once you do that, you get these little balls that are known as nucleosomes. And remember, I told you that would be important later. Here it is. We're going to come back and talk about that a little bit later. So this is the timeline so far. We still have a little bit more, but are there any questions so far? Okay. Then in the 50s, we discovered that DNA was the molecule that acts as the hereditary material. So that's the decade when we finally figured that out. And once we figured that out, that ushered in molecular biology, which is the study of heredity at the molecular level. So again, this is like what we learned in the last chapter, except in the last chapter, we really focused on the big picture and saying, all right, if this thing mates with this thing, then these are the results, right? A lot of those questions are going to be like that. What are the chances of this happening if this thing mates with that thing? Here, we're really zooming in. We're looking at the molecules responsible for this. So we're going to look at the mechanisms, see how that works. Speaking of the 50s, the next word for attendance is another name. We'll just say Chris, but the whole name is Christopher Boyd. If you guys don't know who that is, it's the professor in Back to the Future. And when they went back to the future, it was in the 50s. So that's what reminded me of that. And the date when they went back to the future was October 21st. I think it was 20th. I don't know. It was sometime recent, like it was one of the past few days. So very relevant. But yeah, anyway, today's his birthday. So Chris is the next word for attendance. Anyway, any questions so far? All right, nothing really important yet. This is all kind of like an introduction. We get like catching you up so you know where we're at before we get into this stuff that's important as far as the exams are concerned. So again, in the 50s, like I said, that's when we discovered that DNA was the hereditary material, right? They put all that stuff together. So once they figured that out, then they really wanted to figure out its structure. Like I said, way back in chapter 1, And since then, I've mentioned it a few times, there's this real important correlation between structure and function of biology. So in all actuality, they kind of wanted to figure out how DNA worked, right? And they knew if they're going to figure that out, first they need to figure out the structure. And once you figure out the structure, very often that can help you figure out the function. So at this point in the 50s, people did know all the atoms that were involved in the DNA molecules. They knew how they're bonded, as in like, all right, this thing is triple bonded or double bonded to this thing. That thing is triple bonded to that thing. There's a single bond here, hydrogen bond there, right? These are things they had figured out, but they didn't understand the three-dimensional arrangement, right? It was unknown. So this is the story we're telling in this chapter, right? For a good portion of it. This portion of it, I'm going to tell you the story of how we figured this out. And again, it isn't trivial. So sometimes it does seem trivial. Like, oh, you figured out the shape of DNA, big deal. But it's not just the shape, it's not just the structure, right? Once we figured out the structure, then we knew how it stored genetic information. Because we, at this point in the story, they figured out that it stored genetic information. They just didn't know how. They knew how it was copied, which they didn't know previously, right? This is all stuff you can figure out once you figure out the structure. And then finally, once they knew the structure, they can figure out how we can pass this information from generation to generation. So again, very relevant to your everyday life. This is one of those chapters that goes beyond just, hey, you need to know this because it's a biology class. This is stuff that you know will help you later. All right, so let's talk about, again, this is the first main bullet point, DNA structure and replication. First of all, we're going to have a little review here and talk about DNA and RNA structure. But you already learned a little bit about this, right? In chapter 2, I think it was, when you learned about biological molecules, there was 4 types of them. One of those types of biological molecules was nucleotides. And I said then, I'm going to go through this quickly, because later in the semester, we're going to talk about it again, and that's when it's going to be important. So now is when it's important, right? There was only a few questions about it on exam 1. But now this is going to be very important, so make sure you're paying attention. This time it is important. So again, as you already know, DNA and RNA are considered nucleic acids, right? That is the type of biological molecule they are. They're nucleic acids. They're not proteins. They're not lipids. They're not carbohydrates. They are nucleic acids. Now, granted, as you know, the chromosomes themselves have some proteins in them. The DNA and the RNA specifically, nucleotides. Just like in the last chapter, remember in exam 1, I said it's going to be important to know the polymer and the monomer. Same here. So to remind you, the monomers are called nucleotides. The polymer is called polynucleotides. That's nice and easy, right? When we're talking about DNA only, and we'll talk about RNA a little bit later, but if we're only talking about DNA, then these are the 4 nucleotides, and you will need to be familiar with A, C, T, and G. Not only that, but later we're going to talk about which nucleotides go with which, and you're going to need to know which goes with which. But for now, this portion of the chapter, I'm going to just remind you that those are the 4 types of nucleotides for DNA, A, T, C, and G. Then again, later, we'll talk about RNA, at which point that'll be A, C, U, and G. But for now, don't worry about it. Any questions about this slide before I show you a picture of this information? All right. So again, this is just DNA. You can see these double strands, right? So basically that's 2 molecules brought together with these hydrogen bonds that we're going to talk about later. You can see each one of these nucleotides that you already learned about, right? Your G, A, C, and T's. You can see how they're bonded together. And if you remember, this is the sugar phosphate backbone, right? It's the sugar that's bonded to the phosphate that's bonded to the sugar, blah, 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 so on and so forth. And again, we'll talk about this in more detail later, but this is the big picture stuff. So any questions about this so far? All right, let's talk about these nucleotides. And again, this is all information that you got before. This is information from chapter 2, but we're going to talk about it again because now it's a little bit more important in the context of the exams, the context of understanding what's the information in this chapter. These nucleotides, this is very important, they are joined by covalent bonds. So it's the covalent bonds that are keeping the sugar attached to this phosphate, which is attached to this sugar, which is attached to this phosphate, so on and so forth. So if you look at a single strand of DNA, remember there's 2 of them, in a DNA double helix, it's 2 strands of DNA. If you look at a single strand, it's these covalent bonds that are holding it together. And if you remember, And I told you this would be important way back when I told you. Now it's coming back up, and it's finally important. Covalent bonds are the strongest of the three bonds I taught you, right? So there was covalent bonds, ionic bonds, and hydrogen bonds. Covalent bonds are the strongest of the three bonds. That will be important later in this chapter. So any questions so far? And again, this is all a review so far. Sugar phosphate backbone. You learned about it in chapter 2. And again, just like, even though it's not explicitly written here, just to remind you, since it's the sugar phosphate backbone, remember nucleotides have 3 parts. It's the sugar and the phosphate that are attached to each other. The bases just kind of hang off like little appendages doing their own thing that we're about to talk about. So again, like you know from chapter 2, like you're going to get questions about this. There's 3 parts of a nucleotide. So again, remember these are the monomers. Nucleotides make up polynucleotides. There's 4 versions of them: A, C, T, or G. If we're talking about DNA. And regardless of which version you have, all nucleotides have these 3 parts: a nitrogenous base, a sugar, and a phosphate group. Definitely need to know that. And of course, as you already know, the sugar and the phosphate group are attached to each other. But anyway, let's talk about these 3 parts in a little bit more detail. A little bit more detail than we mentioned in chapter 2. So here we go, the nitrogenous base first. Again, it kind of projects from that sugar phosphate backbone, right? It's kind of doing its own thing. It's not covalently bonded to another thing. Well, it is covalently bonded to the sugar, but you don't need to know that. The point is, it's kind of hanging off, right? It's not a part of that backbone. Here's another thing you don't need to know. It's the second bullet point. It does have a ring of carbon and nitrogen with various chemical groups attached. But for us, that's not important. I only included it because the textbook included it. It is basic, as in not acidic, hence its name. But again, I'm not going to ask you about that. And yes, it does come in 4 varieties. So again, for DNA, A, T, C, or G. And to be clear, when we're talking about the 3 parts of nucleotides, there's 4 different versions. It's the nitrogenous base that defines which version it is, right? So whether you're talking about an A, T, C, or G, or U, if you're talking about RNA, it's the same sugar. It's the same phosphate group. The only thing that makes one of those 4 different from each other is that nitrogenous base, right? That's the only part that's different. So a T has a different nitrogenous base than an A, but they have the same sugar and the same phosphate. So again, nitrogenous base, very important. Any questions about nitrogenous base? All right, next is the sugar. We basically just need to know that there's sugar, but I'll talk about it a little bit more. And I will put a big old X through that whole thing. It's in the textbook, so I'm including it, but I'm not going to ask you any of these questions. The sugar does have 5 carbon atoms, so it's not like some of the other sugars you've learned about, right? Like glucose, for example, as you know, is C6H12O6, right? So 6 carbon atoms if it's glucose. This particular type of sugar only has 5 carbon atoms. It has 4 in a ring and then 1 above the ring. And if we were really studying this, a little bit more closely, that kind of information would be important, but we're not. It's a 100 level course. We're just skimming the surface. The ring does include an oxygen. And when we talk about it, like when you have biologists and chemists talking about this, they have a way of naming things. So the carbons are numbered clockwise from the oxygen, like 1', 2', 3', 4', 5'. But again, you don't need to know that. It's in your textbook, so I wanted to include it. That way I can tell you verbally, hey, you don't need to worry about that. That way when you get around to reading it in your textbook, I'm sure you guys are already in your textbook, you can remember it at least saying, oh yeah, I don't need to know all that. So any questions about the sugar? All right, phosphate group. You've learned about this before too. You've got a phosphorus at the center. This is where it gets its name acid, right? The deoxyribonucleic acid for DNA or ribonucleic acid for RNA. This is where it gets its name. It's the phosphate group that's acidic. And there's a negative charge on one of the oxygen atoms, and I'm going to put a big X through that as well. 100 level course, you don't need to, I'm not going to ask you these things. And you really learned about this more than the other stuff, because remember when we talked about ATP, ATP is a nucleotide. It's an A. That's the A in ATP, right? The adenosine. So it's basically just a nucleotide, except it happens to have 3 phosphate groups instead of just 1. And that's when we really talked about the phosphate groups and how they're so negative and hard to push together because they are repelling each other. And therefore, the harder it is to build a molecule, the more energy is released when you break it, all that stuff. So, yep, that's back. Any questions about the three parts of a nucleotide? All right. So, again, there are four different types of nucleotides, and they all differ by their nitrogenous base. So, right, it's the nitrogenous base that defines the nucleotide. It's the sugar and the phosphate. They're the same. It's the nucleotide, the nitrogenous base is different. Now, this is another thing that I'm not going to get into later or in the exam. Even though there's four bases, they're also subdivided into two categories. They're kind of separated by, are they single ring or are they double ring? So yes, T and C are single ring and A and G are double ring. You do not need to memorize that, hence the big X to it. But what you will need to know later is that T's and A's always go together and C's and G's always go together. And just so you know, now that I'm going to ask you this, hopefully it'll help you understand why T's and A's go together and C's and G's go together. Single ring, T is single ring, A is double ring. So it's the perfect width. If you had two double rings together, it would be too wide. If you had two single rings together, it would be too skinny, right? So the fact that one is single and one is double, that is important to understand how this works. But because it's a 100 level class, I want you to memorize the fact that some are single and some are double, and that's how they go together. But I don't require you to memorize that T and C are the single ones and A and G are the double ones. So any questions about that? All right, here's some more important stuff that I've taught you before, and it's coming back. You need to know the differences between RNA and DNA. They have the same chemical structure, except, like I mentioned already today, RNA does not have a T, it has a U instead. So there will be a test question, and here's a term I haven't given you yet, but there will be a test question where I give you a strand of DNA and I say transcribe this, which means take this information of DNA and turn it into a molecule of RNA. Of course, I'm not going to say that. The question is going to say transcribe this, and that's it. And it's going to be up to you to remember that that's going from DNA to RNA. So I'll go ahead and tell you now, one thing that can help you save time when you're taking this exam is when it says transcribe something in this, multiple choice exam, if there's anything with a T in it, go ahead and not, you cross that off the list, because RNA does not have a T. So if I'm telling you to transcribe something, I'm telling you to create a molecule of RNA. So if there's a T in it, that's the wrong choice, right? So that'll help you narrow down which is the correct answer. As a matter of fact, you might even get partial credit. So if you get the wrong answer, but at least you didn't choose one with a T, you might get partial credit. Because at least then you realize, okay, I'm making a molecule of RNA, and T is not part of RNA. So any questions about that little point? All right, that, like I said, that for us is important. What's not important in this 100 level class is that they have different sugars. RNA has a different sugar called ribose. DNA has deoxyribose, hence the names ribonucleic acid versus deoxyribonucleic acid. But as far as we're concerned, it doesn't matter, right? Not for this 100 level class. I mean, I might ask you to know that RNA has a different type of sugar, but you don't need to know which is which. The more important thing here as far as the differences, as far as we're concerned, is that bullet point. You guys should stream this so that way you can watch it while you're on your phones to help you prepare for the exam. All right, the next word for attendance is nut. Today is National Nut Day. Actually, no, that's not the next word for attendance. Tell me what your favorite or least favorite or last nut was that you had.
What?
Today's nut day, so you could say, oh, for the next word for attendance is peanut because that's my favorite nut, even though it's technically a legume and not a nut. Or my least favorite is almonds. Keep it that way. Phil, I know what you guys are thinking. It's not. Anyway, yep, that's the next word for attendance. Let's move forward. If you download this PowerPoint, you can kind of watch this little animated description of what I just showed you, talk about the differences between DNA and RNA. And there are still some other differences that we're going to talk about later. You can also download this PowerPoint and watch this thing about this Hershey Chase experiment. I'm not even going to mention what it is. You can look it up if you want. You can watch this if you want. It's another paper that you can write for extra credit if you want. Or you could just send an email and say, hey, I looked it up and the Hershey Chase experiment was this, right? Give you just like a one sentence answer. Don't forget your source for 1 point. Or again, you can write a paper about it and get more points. All right, any questions? Okay, now we're getting into the important stuff as far as the discoveries are concerned. So remember, I was trying to give that as a story. We were giving a story, and then we kind of went off on the side tangent and started talking about DNA and RNA structure. Now we're going back to the story of what we discovered and how we discovered it and the implications of those discoveries. So we're going to talk about Watson and Crick's discovery of the double helix. So it was Watson and Crick who determined that DNA is a double helix. And like I mentioned earlier, that was important. It might sound trivial to you, but they were trying to figure out how DNA works. And to figure out how it works, they need to figure out the shape. And they did. They figured out the shape. What is a double helix? It's a spiral that has a uniform diameter made up of two polynucleotide strands. I'm probably not going to ask you that, but there you go. That's what a double helix is. And more importantly, when I use the word double helix, you know what I'm talking about, right? I'd say probably the most important thing to this is the fact that when you look at a DNA molecule, right, that spirally thing, just remember that it has two strands of DNA that are together. And that will be important later. Because again, like I said, structure and function is an important relationship here. In this particular case, at this level, this 100 level class, the most important thing about the structure, I think, is the fact that we have two different strands. And we'll talk about that again later in this chapter soon. So the way it happened was there was this guy named Watson. He saw an X-ray image that was produced by somebody else. Her name was Rosalind Franklin. I'll talk about her a little bit in a second too. And then by using wire models, Watson and Crick took all that information and all the information we had about DNA. They were able to put it together and finally construct a double helix model. And again, I know that seems trivial, but it's not. Because once they figured out that shape, they can figure out how it worked. Now, for the exam, it's not a history class. I'm not going to be asking you specifically about Watson or Crick or Franklin, or the fact that Franklin was the one that had the X-ray, and then Watson is the one that saw the X-ray. None of that stuff, right? This is not a history class. Here's a picture of Watson and Crick working on that double helix model. You can look into them for extra credit too. What I like about them is they weren't exactly good students. They were actually pretty crappy students. They just happened to stumble into a huge discovery. They like to spend their time at the bar a lot. They weren't particularly good at research. They weren't particularly good at classwork. They were just kind of mediocre students. And now look at them, probably two of the more famous biologists in the world in the history of biology. And then there's a picture of Rosalind Franklin. Anyway, so the model of DNA, again, now we're still talking about the structure of DNA like we were previously, but now we're talking about it as if it's a story. But the model of the DNA is like a ladder twisted into a spiral, right? So you can imagine that ladder there. It's not twisted into a spiral in that picture, but there's a ladder, right? The sides of the ladder represent that sugar phosphate backbone, right? Those two things that are covalently bonded to each other. The rungs, right here in the middle, that would represent the hydrogen bonds between those bases, right? Those nitrogenous bases are hydrogen bonded to each other. So again, this one strand here, that's covalently bonded to itself, right? This one strand here is covalently bonded to itself. But it's a hydrogen bond that's keeping this strand connected to this strand. And it's these rules that I mentioned earlier that I'm going to bring up again and show you in writing. The base pairing rules that dictate which go with which. A's always go with T, C's always go with G. And that will be important as we move forward. Any questions so far? All right, like I said, you got a twisted or ladder, you twist it, and that's what a double DNA double helix looks like, right? And again, for us, the fact that it's twisted is not very important. What will be important is the fact that this strand is hydrogen bonded to this strand, right? It's hydrogen bonds that are keeping the two strands together. And it's covalent bonds that's keeping an individual strand together, right? So this strand, again, it's a covalent bond keeping this strand together. Covalent bonds keeping this strand together. Hydrogen bonds keeping this strand together with this strand. Here's some pictures of what I'm talking about. It's the same thing, different drawings, right? So you can see, again, all these hydrogen bonds, right?
The sugar phosphate backbone, they draw it as a solid piece because it basically is, right? Because those are covalent bonds. But then you can see there's these little dashes between the nitrogenous bases because those are hydrogen bonds. Very well, it's a very great picture that indicating what we're talking about here. Another thing that you don't need to worry about, but I'm at least pointed out, notice that A's and T's have 2 hydrogen bonds together, and C's and G's have 3 hydrogen bonds together. That is not for us, it's not important. But for somebody who's really studying this, that would be important. But yeah, but that's just another way they're different. Because remember, I told you too that double rings and single rings always go together, right? Double ring, single ring, double ring. So that's one of the way that the molecular machinery can tell what goes together. And then the other way is by these hydrogen bonds, right? So an A is not going to have even though it's a double bond, or excuse me, double ring, then A is not going to be put together with a C. Because an A has these 2 hydrogen bonds, C's have 3 hydrogen bonds, so they're not compatible. For 2 different ways, they're not compatible, right? First of all, a double in this case, the double and a single is compatible, but 2 hydrogen bonds versus 3 hydrogen bonds is not compatible. Any questions about that slide? Okay, and to reiterate before I move forward, the fact that they have 3 versus 2 hydrogen bonds is important for the physics of this, but for this class, that's not important. And what's definitely not important for you guys is you definitely don't need to memorize the fact that G and C have 3 and A and T have 2. Just keep it in the back of your mind. So again, when you're wondering, when I'm teaching you at the molecular level how we're copying the DNA and how these machines work, the cellular machines, just know in the background, this is part of it, right? Single rings don't go with single rings. Double rings don't go with double rings. Things that need 3 hydrogen bonds do not go together with things that need 2 hydrogen bonds. If you if you download the PowerPoint, this is another little video that you can watch. It's a short video that talks about the DNA double helix. All right, finally, in 1953, Watson and Crick published a paper in a peer-reviewed journal that proposed a molecular model for DNA. And again, that was a really big discovery. I'm going to put it next to it because, again, for the history of biology, it's important. But for you studying for your exam, that date, those names are important. The fact is, the important thing here is that they finally discovered the molecular model of DNA. Then finally, almost 10 years later, they got the Nobel Prize for it. Unfortunately, Franklin was not alive at the time. She had died prior to that, so she didn't get a piece of the award. But yeah, they won the Nobel Prize for that. If you click on that picture of the Nobel Prize, if I remember correctly, I think it's a little video. Yeah, I don't remember. But anyway, that might be a link to a video that you can watch that talks a little bit about their award. But again, as far as we're concerned, not very important for the exam. This is a little bit more important here, what we're about to get at. So once they figured out the shape, right, the structure of DNA, they wrote that it immediately suggested a possible copying mechanism for the genetic material. So again, right away, they were basically like, oh, that's how it works. As soon as they figured out the shape and the structure, they figured out, okay, this is probably how it works. And this is probably how it's going copied and going from one generation to the next. It's the structure of the DNA that points towards the molecular explanation for its unique properties. And that's what we're about to talk about. Finally, we're getting into DNA replication. So this is all the stuff that you learned about. Well, I take that back. Previously, you learned that DNA replication happens in the S phase of interphase. We just left it with that. Now we're going to talk about how it works. So as you already know, when cells reproduce, they have to duplicate their information, right? They have to duplicate everything. And what we were focused on in the context of mitosis and meiosis, we were focused on those chromosomes. And in a sense, we still kind of are. But now we're not just talking about turning a chromosome into 2 sister chromatids and separating them. Now we're really focused on specifically making a good copy of that chromosome, right? It has to be a perfect copy. You want to pass along the perfect copy to the offspring. So it was Watson and Crick's model that suggested how that was going to work. So let's talk about that. Finally, after we talked about hydrogen bonds and covalent bonds, now it's finally going to be making sense if it hasn't already to you, if you haven't already figured it out. So what we have here is the original strand of DNA. What's happened is it's been opened up, right? And then these bases help dictate the copy of the new strands. So the important thing here, like I mentioned earlier, it's covalent bonds that keep that sugar phosphate backbone together, right? But it's hydrogen bonds that keep the two strands together. And remember, hydrogen bonds are the weakest. Covalent bonds are the strongest. So once they figured out this structure up here, it was kind of clear to them, okay, this is how it works. This is how it must work. That thing must open up because those hydrogen bonds are weak, leaving these bases exposed. And therefore, there must be some sort of cellular machinery that comes in and reads it. It says, okay, here's an A, bring me a T. Here's a C, bring me a G. Here's a G, bring me a C. And they figured out pretty much right away that that's how it must work. That's probably how it works. And they were correct. That is how it works, right? It's reading it. The cellular machinery that I'm going to teach you about later. It's opened everything up and it's reading the code and it's bringing in the correct nucleotide to help build that new strand of DNA. Another thing, there will be a test question, and I'm trying to remember how it was worded, but basically, keep in mind that when we duplicate DNA, the two new daughter strands, or the two new double helixes, right? We have one double helix, right? Then we go through S phase of interphase, and now we're left with two double helices, right? Now, what do the two new double helices consist of? They consist of one parental strand, right? One old strand and one new strand. That will be a test question. So make sure you know that. And hopefully that makes sense, right? Because we're opening it up and we're using that old strand as the template to build the new strand. So hopefully that's very intuitive that each new double helix is going to consist of one old strand and one new strand. Because that's how it works, right? Again, it's the old strand, the bases of the old strand that are dictating how we're going to build the new strand. So any questions about this slide? Okay, if you download this PowerPoint, you can watch an animation of how it works, and it moves really quickly. Also, if you do that, if you're watching those little videos that I've embedded in the PowerPoint, if there's any words you don't recognize, and then you look at your textbook to see what those words are. If they're not in your textbook, don't worry about it. So, yes, because these animations were built for a little bit higher advanced students, not 100 level non-biology major. But anyway, you can watch that. You can also watch this. This one is more along your speed, so that one's probably not going to have any terminology that you don't need to know. You can download it and watch it if you want. Here's yet another one, and this is a great example of some words that I'm not going to teach you about. For example, we're not going to talk about template strand or lagging strand. I'm not going to use the word primase. Yeah, there's a bunch of stuff. So when you watch these videos, if there's a term you don't understand, you can just kind of ignore it. Or again, double check on your textbook to make sure it's not a word that you do need to know and you forgot about. Anyway, here we go. Here's the first term that you need to know. I think the first new term, right? There's some other stuff you need to know, but this is the first new thing I think from today that you need to know. DNA polymerase. You need to know what it does. As you already know, enzymes are named. The end of the name is ACE, right? ASE. And they're usually named after their substrate or what they do. So hopefully that makes sense. DNA polymerase, that's the enzyme that makes the DNA polymer. So that's the cellular machinery that's doing what we talk about in S phase of interphase. It's the thing that's opening up the DNA to read the code. It's the thing that's bringing in these new nucleotides to build a new strand. Right? It's DNA polymerase. Just to skip ahead, just so you know, for the exam, there's going to be two enzymes you need to know. And hopefully you'll get this right because it's in the name. DNA polymerase makes the new DNA. The other one that you're going to need to know about that I'll tell you about officially later is RNA polymerase. And guess what it makes? RNA. Perfect. Yes. So yes, for the exam, there'll be a test question. Don't get those two confused. DNA makes DNA polymers. RNA polymerase makes RNA polymers. So again, this is the cellular machinery that's taking the incoming base pairs and making sure it's the correct ones. Like, all right, here we have a double base pair on the old strand, the parental strand, so or a double ringed nucleotide. So now I need a single ring nucleotide. This one has 3 hydrogen bonds. Okay, so this one also needs 3 hydrogen bonds, right? This is how it's determining which ones are going with which. It's not like they have the letter A or T written on them in your cells, right? This is how it's working at the molecular level. It's looking at the size, it's looking at the hydrogen bonds, putting them together. Here's a number I might not ask you, but I'm not quite sure yet. But it is fast and accurate. Specifically, it does about 50 nucleotides per second. That's how fast it is. And how accurate is it? There's usually 1 in a billion bases that are incorrect. So it does it all the time. And once in a billion cases or bases, either one, it'll mess it up on average. So that's what DNA polymerase does. It builds the new DNA strand. It's also worth noting that not only does it build new DNA, it also helps repair old DNA. Right? So when you get something happening to it, because maybe you're smoking, or maybe you've been out in the sun too long, whatever it may be, whatever it is you did to damage your DNA, DNA polymerase can help it. Of course, as you know from our discussion about cancer, it must not be perfect, because if it fixed all your mutations, nobody would ever have cancer. And as you're going to learn later when we get into natural selection, obviously if it was perfect and we didn't have any mistakes, then we wouldn't have any different alleles, right? All genes would be the same, therefore there would be no natural selection. So no, this stuff is not perfect, but it's pretty accurate, pretty fast. So any questions so far about DNA polymerases? All right, another thing I want to mention, and I don't remember if your textbook does or not, is this is an oversimplified explanation. DNA polymerase, it almost sounds like I'm talking about one enzyme. In the context, the way we're describing it, it is one enzyme. But in all actuality, what's happening is it's a team of enzymes working together. So if you were in a biology class as a senior, you would be learning about those team of enzymes and how they work together in these different proteins. But here, we're just oversimplifying it and saying all of them together are called DNA polymerase. And I only mention that because, again, hopefully one day, as critically thinking, college-educated citizens, you're going to be presented with some information. Instead of just believing it, you're going to say, oh, and I'm going to look that up. And you're going to look it up. And then you might find some words that you don't recognize. Right? I thought you said it was just one enzyme. So now I'm telling you, yes, we're calling it one enzyme, but it's a team of different proteins. Anyway, so again, DNA polymerase, that's the machine. You can't see it in here, but it's the thing that has pulled those strands apart. It's the thing that's reading that G or that C and bringing in a G and, covalently bonding it here. It's the thing that's reading that C and bringing in another G and covalently bonding it here. It's the thing that's going to read that A and bring in another T and covalently bond it here. Okay, that's the cellular machinery that's doing all that. So DNA replication, as you already know, happens during S phase of interphase. Obviously, you're going to get a question on that because I've taught it to you in 2 different contexts. Well, I guess sort of 3. First of all, when we talked about cellular reproduction and mitosis and meiosis, we said DNA replication happens in S phase of interphase. You needed to know it then. You need to know it now. It starts at these places called the origins of replication. I'll put a question mark next to that because I'm not quite sure if I'm going to ask you that. In other words, what it doesn't do, because this is how I would have thought about it if no one taught me. You told me a machine opens up the DNA and copies it and makes new DNA. I would have figured it started at one end and worked its way to the other, but it doesn't. It actually starts at different places. Throughout your long strand of DNA, there's multiple origins of replication. That's where it attaches, and it kind of works its way out from its attachment point. And that one's also, this would be better if it was animated, right? Imagine that bubble getting bigger that way, and this bubble getting bigger this way until they finally meet together, and there you go. Now you have 2 new daughter DNA molecules, 2 new double helices. So any questions about this slide? All right, download the PowerPoint. You can watch it in action because, like I said, that would be better if it was animated, so you can watch that animated if you want. Same with that. Again, we're not going to talk about lagging strand versus leading strand, but it'll be there. Again, if there's any terminology that you don't recognize, don't worry about it. Like 3' to 5', or 5' to 3', things you don't need to worry about. If the word doesn't make sense to you, and then you look it up in your textbook, and you don't see it, then that's your cue to brain dump it. All right. So here, again, this is the same, the second main bullet point of our 3 main bullet points of this chapter. And as far as the exam is concerned, I would say this is probably the most important. This is where you're going to get most of your questions. And to understand how viruses work, you need to understand how we go from DNA to RNA to protein. To understand how mRNA vaccines work, you need to understand how we go from DNA to RNA to protein. For most genetic diseases, you need to understand how we go from DNA to RNA to protein. That's, if it's a genetic disease, that's exactly what we're talking about. If you have a bad allele, that allele is going to dictate how to make RNA. That RNA is going to dictate how to make that protein. And if that protein is faulty, you then have that disease, like some of the diseases we talked about earlier, like Huntington's disease or sickle cell disease, all these things. So let's jump into it. Actually, before we even do that, this is not one of those times I want to remind you, everybody learns differently, but I would recommend studying the big picture and then adding the details. So for example, the first thing you should memorize is this. We go from DNA to RNA to protein. And that'll also be a test question. It'll say which one of these is the correct order. DNA to RNA to protein. Memorize that first. That will be a test question. So it's not RNA to DNA to protein or protein to RNA to DNA, right? It's DNA to RNA to protein. Make sure you know that. Then later you can add some details. Like there's a name where we go from DNA to RNA. You're going to need to know it. For now, I wouldn't worry about it. And there's a name for where we go from RNA to protein. You need to know what it's called. There's specific enzymes involved in going from DNA to RNA. You need to know them. There's specific enzymes between going from RNA to protein. You're going to need to know them. But again, for now, big picture, DNA to RNA to protein. Any questions about that? All right. So, as you already know, but here it is again to remind you, DNA provides instructions to a cell. That makes sense, right? Your cells basically runs on proteins, right? Even when there's something else involved, like a carbohydrate, for example, it's still a protein that helped make that carbohydrate. Because remember, enzymes are made up of proteins as far as we're concerned. There's a few exceptions, but as far as we're concerned, it's always proteins. And remember, proteins are enzymes and enzymes are proteins. And excuse me, let me make sure I'm saying that right. Enzymes are proteins, not always necessarily the other way around, right? Not every protein is an enzyme. But my point is, all these chemical reactions, as you learned in the previous chapter, are all due to enzymes. Without enzymes, you wouldn't have these chemical reactions. So yes, there's a lot of stuff going on in your cells that aren't proteins, right? We've got lipids and carbohydrates and stuff like that. But the point here is that without these enzymes, we can't make those other things. So yes, it's not as simple like proteins aren't everything necessarily, but proteins, these enzymes do dictate or help build these other things. So again, without this, that wouldn't work. Yeah. So the question is, what you're going to learn is how does the DNA provide instructions for all these different proteins? And that's what we'll answer when we come back on Friday. And that's it. We're done for the day. Don't forget to, your last word for attendance is to send me a question about the material presented in this. Make sure you get it sent in by 9. And I'm staying here. You guys can leave if you want, but I'll be in this room if you need me for lab. Don't forget to get those words in by 9 a.m. please. Right.